change in genetic sequence? So your answer is very precise, but just to, do you think if we found someone on Pacific Avenue, we asked that, they would come up with that? It was one of you guys, yes, if we happen on one of you guys. But we're probably not gonna wind up with one of you guys. So if we just saw some, like, not scholarly person like yourself, just a random, regular person out there, what do you think they're gonna say? A mutant is. Okay, maybe a creature. Defective, yes. Anything else? So something not like normal, not like the normal. What were you gonna say?
X-Men.
Okay, X-Men, yeah, could be, yeah. Anything else? Is there still such thing as ninja mutant turtles or mutant ninja turtles? Yeah. Okay, so those turtles, are they normal turtles? No. Is there something wrong with them? Not exactly. I mean, they're not like a normal turtle. They're like a superpowered turtle, would you say? So usually, mutation, as was mentioned very correctly, is a random change in the DNA. So normally a random change in the DNA, you wind up with the wrong amino acid in that protein. So do you think that's usually going to be good? No, it's going to make it defective. So that's why when you think of something as a mutant, you think of something not the normal, whatever that thing is. Occasionally, however, that something might be better than it was, and then you get an X-Men or the Ninja Mutant Turtles, and that can happen out in nature too. So mutation is part of allowing for genetic variation over time among all kinds of things, whether it's viruses, bacteria, people, etc. So we have a lot of genes, so does E. coli and other organisms. Do you think organisms are expressing all their genes all the time? Yeah, that would take like a lot of ATP, because that would take a lot of effort to unwind your DNA, unzip it, get all the enzymes involved and make your messenger RNA, transport that over to the ribosomes and all of that going on, a lot of energy. So you don't want to just be expressing things that you don't need. So there's a lot of regulation involved in expressing your genes. And if you go on in biology, you'll probably take a course where you'll learn all about all that we know, which is amazing, all that we've learned over time. I'm going to tell you one way of gene regulation, and that's called the lac operon. This is one that ties into things we've already learned about. This is found in E. coli. This is used by E. coli. These are genes that are involved in letting E. coli use lactose as an energy source. So there are actually 3 genes that are needed that make 3 different enzymes that allow lactose to wind up in glycolysis. Those enzymes are listed there for gene 1, gene 2, and gene 3. Of course, these are the DNA that codes for making the protein, which is that enzyme. And this is the DNA that codes for making the protein, that is that enzyme, and so forth. So these things here aren't the actual proteins. Those are the genes that tell the cell how to make those proteins. The cell will need all 3 of those to get lactose into glycolysis, to use lactose for energy.
I think if you have the gene, right? Like if you did not have the gene for it,
Then none of this would happen. Right. And we're focusing just on this particular scenario, which is happening in E. coli. And yeah, so this is different than what would happen in us.
So that microorganism having the gene for the lactose.
For breaking down lactose. Right. genes in order together, because if you need one of them, you need all three of those. So they need to be expressed together, so they're regulated together. When we transcribe genes, there's a promoter region that's at the beginning of the gene, or in this case, the whole set of genes. That's where our RNA polymerase binds and then goes along there and makes the messenger RNA. These genes, though, are normally turned off because there's a regulatory site called the operator in between the promoter, which is where the RNA polymerase binds, and the actual genes. It's turned off because there's a teeny tiny gene here, regulatory gene, that makes this teeny tiny protein, and it always makes this protein. So this thing gets stuck right there. So these genes are turned off. So the normal situation is these genes are turned off. When would E. coli need to turn these genes on?
And lactose is its only energy source?
Yeah, if it finds itself in a big glass of milk, a lot of lactose there, so now it needs to express those genes so that it can eat the lactose. On this repressor protein, there's a regulatory site here for something to repress the repressor and make it let go of being stuck there. What do you think the logical thing is for what would get stuck in there to inactivate the repressor? Lactose, yes, because when lactose is around, E. coli needs to express those genes. So if lactose is around, it binds to this spot. So this thing is stuck there, lactose comes in there and binds to that part of it, and it changes its shape and makes it let go. And now RNA polymerase can do its thing and make the messenger RNA for all three of these genes. Then we get a translation. E. coli gets to eat the lactose. And if it happens to be in your, so we related this a little bit in lab to our own situation. We have good E. coli in our GI tracts. All of us have good E. coli right now. It's making vitamins for you. It's helping digest your food. And if you're not able to break down your own lactose, because mostly usually babies can break down lactose because that's in their mother's milk. But then sometimes people lose the ability to do that as they get older, and so then they're not breaking down the lactose. So if you drink a big glass of milk, the E. coli that's in your GI tract is going to break down the lactose for you. Our GI tract is anaerobic, so that no aerobic respiration is going to happen. And the E. coli will turn on these genes, break down your lactose, and carbon dioxide is one of the products that it makes when it's fermenting the lactose, as you guys saw. Remember the durum tube when we did the lactose fermentation test? That durum tube was like full of gas. And that's what happens in your GI tract if you aren't able to break down your own lactose. So if you find yourself farting after drinking a glass of milk, that is happening because your E. coli's genes are getting turned on by the lactose in there to make the genes, or express the genes that allow it to eat the lactose. And when all the lactose is eaten up, gets turned off, you're done farting, and you decide you're not drinking milk ever again. But if you can digest your own lactose, then you break it down before it gets to the part where E. coli is in your GI tract. Yeah.
So lactose binds to the operator, or where does it bind?
To the repressor protein. Yeah. The repressor protein is bound to the operator, and that's what keeps these off.